Hi, Dr. Cobb. This is Drew Norton doing my glucose transport and insulin video. First, we're going to start out with glucose transport in adipose and resting skeletal muscle cells. So we're going to start out in the fasted state of adipose or skeletal muscle cells. You can see here we've got the extracellular fluid and the inside of the cell. We've got glucose molecules here and an insulin receptor here. Because we are in the fasted state, the pancreas is not stimulated. The blood glucose level is low, therefore the pancreas is not stimulated to produce insulin. And because of that, there are no GLUT4 transport proteins on the membrane of the cell, which means that no glucose is getting inside of the cell. On the flip side, if you're in the fed state, the blood glucose level is high, which means the pancreas is stimulated to produce insulin. So we've got insulin bound to the insulin receptor of the adipose and or muscle cell. This stimulates a signal transduction cascade beginning with the insulin receptor substrate being phosphorylated, and ends with the let's see if I can focus in there, sorry, the GLUT4 transfer protein, the secretory vessel containing them on the on its membrane, that secretory vessel fusing with the membrane of the cell, and that GLUT4 transport protein now allows glucose to enter the cell. So moving on to the glucose transport in liver hepatocytes. Sorry, I know I'm not the best artist here, but work with me if you can. Again, we've got the extracellular fluid. We've got very low insulin, so it's not binding to the receptor here. We've got very low glucose concentration in the extracellular fluid. And because of that, because we're not getting the glucose from the diet at the moment, the liver is in the process of secreting glucose into the extracellular fluid in the bloodstream by way of or from the glycogen storage and from glyconeogenesis. So yeah, we've got these GLUT2 transport proteins, which are bidirectional. They can transport glucose into the cell and out of the cell. Those are on the surface of the hepatic cell, and the glucose is going from a high concentration to a low concentration, as you would expect, from the hepatocyte into the extracellular fluid. On the flip side, we've got the fed state of the liver hepatocyte. So now, because blood glucose level is high, insulin is being produced by the pancreas, and when that insulin reaches the receptor of the hepatocyte, goes through again a signal cascade, whereby it triggers glucose to enter the cell, because, sorry, actually I need to back up. The glucose level inside the cell. So because we're in a fed state, the liver cells are not producing glucose because we're getting the glucose from the diet at the time. So the glucose level inside the cell is low compared to the glucose concentration outside of the cell. So this leads to diffusion of the glucose cells glucose molecules into the liver hepatocyte, and the liver hepatocyte then begins to either use the glucose or store it for future use whenever we are in the fed state. And lastly, you asked what happens in the absence of insulin. My answer to that is that cells are unable to uptake glucose and will have difficulty meeting its metabolic needs and will likely die. There will be a lack of energy storage in the form of glycogen and fat, and there'll be a lack of utilization of amino acids. And this is going to be diabetes. Let's I can't remember which type that is, actually. Let me look that up. Lack of insulin, diabetes type. Diabetes type 1. Sorry about that. I hope that's okay. Thank you for a great semester, and look forward to taking the final here next week.